Everyone. So I wanted to focus this video on the cell cycle and the two bigger phases of the cell cycle, Anaphase and the mitotic phase. So as we can see, there's a lot of um, the cell is in Anaphase for a lot longer than it is in the mitotic phase. It's only in it for this fraction, and the rest is Anaphase. And so this in Interphase, it's just preparing, growing the cell, and just getting ready for division and carrying out the normal functions of the cell. So first in Interphase, we have G1, and so that's gathering everything that the cell needs before it can go on with replicating the DNA, which will happen in the S phase. And this also just like grows the cell if it needs to grow anymore. Then we have S phase. And so this is where we duplicate all of our DNA, and we also make our sister chromatids. They're made and stuck together, but they haven't been condensed quite yet. And we also duplicate our centrosome, which will be needed for all of our other phases of mitosis. And the last part of Anaphase is G2. And so in G2, we basically just make sure all of our energy reserves are good to go. We'll have enough energy to get through the entire mitosis phase, because if we don't have enough energy, there's like no stopping. We can't stop in the middle of mitosis. So we want to make sure we have everything we need. One thing my professor would say is, imagine if you were like going camping in a place in the middle of nowhere and there's not going to be anyone there. You're probably going to make sure that like you have everything you need so you're not stranded in the middle of nowhere by yourself. So that's basically what G2 is. And once we finish that, we can get started with the mitotic phase. And so this is consisting of prophase, metaphase, anaphase, and telophase. Um, in this picture that I have, we do, oh, I crossed out the wrong one. In this picture that I have, we do have pro-metaphase, but you don't need to know that. I just included this picture because I really like the way it looks. I feel like it simplifies everything down a lot so it doesn't look too overwhelming. It does look a little overwhelming, but that's just because mitosis is a really difficult process. So, and a lot of students don't understand it just because of how many steps there are, but we can work through this together. So the first stage is prophase. And this is when our DNA starts to condense and our nuclear envelope, um, which is this thing, it starts to come apart. And then we have our centrosomes, which are these big things, and they've already been duplicated. They start moving to the opposite sides of the cell. So this one is going to start moving here, and this one's already over here, so it can just stay there. And we also can kind of see our mitotic spindles being formed. Just a little, it's not fully formed, but they're starting to form. And these are made from microtubules. Okay, I'm gonna skip pro-metaphase and get to metaphase. But before that, I wanted to talk a bit about the sister chromatids. So I do have this picture here. And so in our sister chromatids, we have, this doesn't show it, but we have a centromere right in the middle. And that is where the two are attached. And we also have kinetochores, and that is where the microtubule or mitotic spindles attach to the chromosome, or the sister chromatids, I should say. And we have both the centromere and the centrosome in the cell. So here is the centromere, here is the centrosome. One thing I like to know, to remember it, is centromere has an M for being in the middle of the sister chromatid. So that's the point of binding right there. I don't know if you can see my arrow, but right there. While the centrosome doesn't have a M, so it's not the middle of the cell, or middle of the sister chromatids. Okay, so now let's get started with metaphase. So in metaphase, this is where our chromosomes or sister chromatids start to line up in the middle of the cell. So as you can see, they're kind of forming a straight line, and that is because both the centrosome on either side are attaching to the sister chromatids at a point. So this centrosome is attaching to this kinetochore. This centrosome is attaching to this kinetochore. And they're basically playing tug of war with the cell or with the sister chromatids. But because they're both have equal strength, the sister chromatids basically just line up in the middle of the cell, creating this line called the metaphase plate. It's not an official place, like a structure of the cell. It's just kind of what it looks like. It looks like a plate in between. We also check to make sure that every single sister chromatid is attached to two different microtubules or mitotic spindles in this phase, just because if it's not, then one of the cells will take both of the sister chromatids. So they'll both go over there, and then that one will have too many copies of a certain chromosome and the other one won't have any copies of it. So we don't want that to happen. So we just do a check, make sure everything is good to go. All of our sister chromatids are attached to two different points. And then once we have finished our check, we can get started with anaphase. And in anaphase, this is basically just an enzyme comes in, cuts between the middle of the two chromatids to let the daughter chromosomes, which is what it is now called, free. And so the daughter chromosomes aren't attached in the middle of the middle in the binding point. So they are free to go to their respective side of the cell. And this is a very fast process because imagine, like I was saying with my tug of war example, if you had a piece of rope and imagine someone just came and took a pair of scissors and cut the rope in the middle, you're probably gonna quickly like fall back. So that's kind of what's happening here. They're quickly going to the side of the cell that they were attached to. And then once this finishes, once they're all on their sides of the cell, we can get started with telophase and cytokinesis. So in these phases, so it's basically just the cells are starting to form, come apart, so they're not bound to each other anymore, and our nuclear envelope starts to reform, the DNA starts to decondense, and everything starts to go as it was to create two new daughter cells. And that is pretty much all of mitosis. We do our most of our checking in metaphase because we want to make sure that all of our cells have the correct number of chromosomes. If not, then we won't be able to have our correct gene expression in the cells, so that might alter our function. Please just let me know if you have any questions. I'm going to create a separate video on meiosis as well, so you can look at that.